Hi all. Let's get back to part 2 now of cells, and I'm backtracking for a couple just so that you can see what the cell membrane actually looked like. So we had proteins inserted in a lipid bilayer. In that lipid bilayer, we said this outer ball region is the part that is hydrophilic because it needs to get along with all this watery extracellular fluid. And the balls on this end need to be hydrophilic also because they have to get along with this watery intracellular fluid. We had glycolipids, which are these sugars that are attached onto the lipid. We had glycoproteins that are attached onto the proteins here. And we had cholesterol in blue here, giving that membrane greater flexibility. So that's a really nice summary there. And we did a clicker. And let's continue on to looking inside of the cell. Okay, so we're looking indoors into the cytoplasm. And in there, there's a very thick fluid that contains all those particles, all those suspended particles and organelles. And that is the cytosol. Cytosol. And the organelles are the little organs. L is little organ, and those are all going to perform very specific functions that we need to go over. So here's Joe's cell again with all its different little organelles that are not living on their own because at the cell level is what living is actually determined as living is the cell. Okay, so contents of that cytoplasm are going to be water, mostly water, right? 75 to 90% water in the cytosol. And the rest are going to be those solid components. So what's the function? Well, really it's where all the chemical reactions are occurring. So it's just a fluid interior where chemistry is happening. That's how we're going to say it. What's in it? Well, what's in it? It's high in potassium. Those are positive ions. High in proteins. Lots and lots of proteins. So many we don't even know what they all are. Those are negative. High in lipids and some carbohydrates. Okay, let's keep looking. It has a skeleton. Yes, it has a skeleton. And I'm not going to go into a lot of detail of this other than we think of it as just being this fluid middle, but actually it's got these proteins that sort of go from one membrane to the other. We have some that are like railroad tracks here. We have some that are larger than others. And so when you see the term intermediate filament, microtubules, and microfilaments, those are all making up this inside skeleton. Some of them are like this, some of them are like this, and then some of them are temporary. And they're only there during cell division. That's a really good example during cell division. So intermediate filaments, microtubules, and microfilaments, those are all forming a skeleton that does give it somewhat of a shape. Which of the following is true regarding HeLa cells? Oh gosh, that was from the last one. Have a huge capacity to divide. Are named after Helen Lawrence. Are still alive today. And D, A, and C are correct, which is right here. And if you said D, A, and C are correct, you are right. All right, let's look at the surface. On the surface, you have villi. You could have villi. You don't necessarily have them. You could have villi, which are extensions of the plasma membrane. So it's the actual membrane come up. Membrane comes up. So the actual extension of that membrane are the villi. Why do you want villi? Why do you want that extension of the membrane? To increase absorption. You've increased the surface area of the cell. Very important for absorption. So in the intestine, I say it looks like a shag rug. It looks like a shag rug because you have membrane up, membrane up, membrane up, and every single location along those villi are capable of absorption of nutrients. What if you don't have villi, but instead you have cilia? So cilia are these extensions that you have that are formed from microtubules. What were they? They were part of the skeleton, remember? Part of the skeleton of the cell. And if they protrude out beyond that, they could form little movement molecules. What they're going to do is trap food or trap something that you don't want in that particular location and move them along. So they're projections. They're not a fold. They're a stick out, essentially. They're short. There's lots of them. They can cause currents on the cell surface and move things like the wave in the stadium and remove things that shouldn't be in that location. So the classic example is in the trachea. In the respiratory, all your respiratory airways that want to trap any particle that should not be in there, then you can blow air, which means cough, and expel that little particle that's sitting there that shouldn't be. We think that some of them might be sensory, but we don't know that yet. We haven't fully understood that, about some of those being sensory. Okay, what other extension can come out from the cell? And that is a flagella. We have villi, cilia, and now a flagella. Much less common, not common. They're also part of the skeleton. It's a microtubule. And they are few. They have usually one, very long. They can move the entire cell, make the cell go somewhere else. And the classic example of that, of course, is a sperm cell that has this flagella that allows it to move and fertilize an egg. All right, so let's look at some organelles, see what you remember of all this. They perform very specific functions. So we're going to look at the organelle and then talk about its function. They're all in here, Golgi and ribosomes and mitochondria and all sorts of good things. So let's start with the ER, not emergency room, but endoplasmic reticulum. So the endoplasmic reticulum is like this flattened membrane that stretches around. And this flattened membrane might be a bumpy membrane or a smooth membrane. So it's called the rough endoplasmic reticulum and the smooth endoplasmic reticulum. What makes it rough is the presence of ribosomes. The presence of ribosomes. So they are attached onto the rough endoplasmic reticulum, and they're scattered throughout the cytoplasm. And what you need to know about that, what you need to remember, is that they are the site of protein synthesis. That's where protein is manufactured. Very important. Smooth ER. If it's smooth, we have no ribosomes. If there's no ribosomes, the function changes. So what it does instead is it synthesizes something also very important, lipids and steroids. Steroid is a lipid. What else? The smooth ER detoxifies in cells. It detoxifies alcohol and other drugs. And it's interesting to know that for an individual that needs that detoxification, right, that is consuming vast quantities of alcohol, then their smooth ER is going to be very present. They can have greater quantity of ER to try and solve that problem of detoxifying the alcohol. The liver, right, would be an organ, that is packed with the ER because that's what its function is. One of its many functions. So when you talk about tolerance to alcohol and drugs, it's because an individual is able to process that more quickly. So an individual who drinks a lot is able to detoxify more quickly and continue drinking. What a thought. Let's switch to mitochondria. So this is what a mitochondrion looks like. It kind of looks like a big bean, and inside it's got all these walls, right? And those walls are called cristae. And what's in between the walls is matrix. So cristae and matrix. And on those cristae are hundreds of enzymes that are going to allow for the manufacturing of ATP. But let's get to that, right? Just think of walls and in between. In between is the matrix. We call it the powerhouse of the cell because it's what's going to generate power for you. It's what's creating your ATP. So it's producing adenosine triphosphate. This is your fuel. That's your fuel for you to move, think, see, hear, everything. You need ATP. It has its own DNA, this mitochondria, which is very interesting. It's a whole different story of inheriting maternal DNA from a mitochondria. That's very interesting. And it's, as I said, a double-walled, has its cristae and its matrix. And that's all we want to know. What do mitochondria do? They produce ATP. They're going to take oxygen and glucose and ADP and manufacture energy, manufacture ATP. This is where cell respiration occurs. What I just described is cell respiration. It's that conversion of glucose to ATP, but you need it to be in the presence of oxygen, where that's how we make most of our ATP. So as a very oversimplification of this, you've had your breakfast, you've eaten your glucose. You're breathing, you've got your oxygen. ADP is the leftover ATP that you already used a phosphate from. So you've got that leftover, and between that leftover oxygen and glucose, you can convert that and slap on another phosphate onto the adenosine diphosphate. It only had 2. You put the third one on. And in the process, you create carbon dioxide, water, and heat. So I always think when I get out of bed in the morning and I feel all that warmth left behind, I think, what's my ATP doing that? Leaving behind all that warmth. This is a summary in case you do not remember what ATP looks like. It's the currency, it's your fuel. It has adenine. It's a nitrogenous molecule, nitrogenous base. There's the nitrogen. It has a sugar, ribose, and then attached onto that is a phosphate, which is phosphorus with 4 oxygens, phosphorus with 4 oxygens, phosphorus sharing oxygens here. This is the phosphate part. What we use for energy then, take my pen, but it's not letting me do that, is one of these phosphates. So that's what I need to power a lot of metabolic reactions in the body. I need that one phosphate. So it donates, this molecule donates, and off you go. You're running, your muscle needs that extra phosphate in order for contraction to happen and for relaxation to happen too. So it's that extra phosphate. So when we're talking about metabolism, we're tagging on that extra phosphate onto this molecule and making that phosphate available for the next metabolic reaction that you need. Whether it be move or think or see or hear, all of that is going to need that ATP. Do one more, Golgi apparatus, and these are flattened bag-like structures, and they are in charge of packaging in the cell. So what they will do is receive some sort of molecule, transport it down in this direction, and form a little baggie out of it. And that baggie then gets exported to somewhere. So it's the UPS. The Golgi apparatus is the UPS. It's packaging and sending it to either within the cell where it needs it or exporting it outside of the cell. I will continue with these different organelles in the following video. Thanks, guys.